So once again, good morning, good afternoon, everybody. My name is Zoltán Perestegi, and as usual, I will be the host of today's webinar. Today's webinar is titled Green Features of Centrifugal Partition Chromatography, and will be hosted by Gergő Dargó, PhD, a research chemist in RotaChrom's research laboratory. But before we jump in, let me give you a quick summary of what RotaChrom is, what we do. We are the world's only integrated industrial scale centrifugal partition chromatography solution providers. RotaChrom is a European company that has presence in all continents throughout the world. And we have a North American operations where we service our customers locally in the US and Canada. With regards to today's topics and for further information about this, we also have a podcast. So the podcast is available on all of the podcast platforms. So go search for Purified by RotaChrom on your favorite podcast platform, and you can download all previous episodes. So the podcast always features the speakers of the webinars and the topics of the webinars. So if you have some time now, go ahead and search for Purified by RotaChrom on your favorite podcast platform. Now, about today's topic and today's speaker. Gergő Dargó, PhD, has been with RotaChrom for quite some time now. He has a scientific background, academic background, and he's also worked in the pharmaceutical industry before joining RotaChrom. So with that, I think I will now hand over and let's start today's webinar with Gergő. Welcome to the webinar, Gergő.
Thank you, Zoltán, and greetings for everyone. Today, I'm going to speak about the green chemistry aspects in the introduction. Then I will jump to the key green research areas. Then we will see some strategies for going green in CPC. And then I will give you a brief summary of these topics of today. So what makes CPC superior to conventional methods when it comes to green chemistry? Well, by changing from the conventional solid-liquid phase separation techniques to a system with liquid-only phases, a significant cost from purchasing the expensive silicate gel and the substantial amount of solvents can be spared. Meanwhile, the generation of solid waste and its disposal cost can also be avoided. Also, in the case of solid-liquid chromatography, we can choose from only a limited number of stationary phases, which can be changed only by the replacement of the whole column. And our options to fine-tune the separation are limited to changing the composition of the mobile phase only. The risk of clogging and irreversible adsorption might also decrease the time interval of this replacement of the packing. On the other hand, in CPC, numerous possible stationary and mobile phases can be used, which can cover a wide polarity range. And the versatility of the system is further increased by the possibility of the interchanging of the roles of the phases during separation. In 2007, the members of the ACS Green Chemistry Institute Pharmaceutical Roundtable sampled the key green engineering research areas, first in the aspect of pharmaceuticals, then in 2011, they extended it to fine chemicals. Here, separation and reaction technologies and solvent selection and recycling were designated as the second and third most important fields of research by the roundtable. Not surprisingly, as separation operations are present in almost every step of the manufacturing process within the chemical industry. These steps are often the predominant contributors to processing costs. Therefore, novel and intensified separation methods are required for a greener and more sustainable manufacturing process. From the aspect of green chemistry, the ecological footprint of a purification process can be reduced by the rigorous management of workup solvents or choosing renewable and green solvents instead of the halogenated and petroleum-based ones. We can consider a solvent green if it processes reduced health and environmental absorption and toxicity, and displays a reduced life cycle impact. To facilitate the choice, several pharmaceutical companies like Pfizer, GSK, Sanofi created their own solvent selection guides. However, as it can be seen from the graph, these different guides rank the solvents based on different criteria. Therefore, conflicting classification have emerged. To remedy this, the Innovative Medicine Initiative, CAM21 Consortium, compiled and combined these previous data to create a selection guide, which ranked the solvents into recommended, problematic, hazardous, and highly hazardous ones. How can we apply this to CPC? From the aspect of green chemistry, several steps of the separation workflow can be appointed as key areas where a significant reduction of costs and ecological footprint may be achieved. First, we can select green solvents as components in the biphasic liquid system. Then the recycling of solvent waste to the start of the separation process also an option. And we can also use the liquid waste as a valorization process as input materials of the CPC. In recent years, several steps have been taken to find new alternatives for solvent system components to create greener solvent systems for CPC. Some of these solvents and additives are shown in this slide. To be considered green, these solvents and additives have to come from a renewable source, have to be relatively non-toxic and easy to recycle, and for safety reasons, it is also preferred if they are non-flammable. Since recent regulations banned several hazardous solvents like N-hexane, which was most commonly used for extractions of natural products, another alternative had to be found. 2-methyl tetrahydrofuran is often considered as a green alternative to THF in synthesis, as 2-methyl THF is a non-toxic solvent. It has low miscibility with water, and the stability compared to other heterocycles is remarkable. Also, it is a biomass-derived chemical, which is in accordance with several of the green chemistry principles. It can be produced from renewable raw materials. It is biodegradable, easy to recycle, has a promising environmental footprint, and also a good preliminary toxicity. If we compare it to N-hexane, as we can see in the table, it has almost the same molecular weight, a higher boiling point and flash point, which makes it less flammable and hazardous. Its higher density and viscosity might be a little bit of a drawback during CPC runs compared to N-hexane, but it is well within the operational range of CPC. Another possible alternative is limonene. As a natural cyclic monoterpene with a pleasant fruity odor, its D-isomer is the major component in the essential oils of citrus fruits. The world production of these fruits is over 120 million tons per year, from which the resulting peel industrial waste is close to 10 million tons. From this biomass, typically 5.4 kilogram of oil per ton can be used as an easily available and renewable solvent for example, CPC separations. From the regulatory aspect, limonene has been lifted by the FDA as generally recognized as safe solvent, and it's already used as a food additive, flavoring, and fragrance additive. On the other hand, it has been ranked as a problematic solvent by the CAM21 guide due to its relatively high boiling point and aquatic toxicity. However, its natural origin still makes it a much better and greener alternative to the alkanes, hexane and heptane, due to their flammability, aquatic toxicity, and the neurotoxic human metabolite of hexane. Limonene and heptane are very close in polarity, as we can see in the table. They also show quite similar solvatation capability towards water and methanol. If we overlap the ternary plots of limonene and heptane with their water-methanol mixtures, we can see that they share strong similarity in the biophysics systems. The most notable difference in their properties is the density difference between the alkanes and limonene, as can be seen in the table, which affects their position in the biophysics systems. In the case of a system with heptane, methanol, and water 10:9:1 ratio, heptane is in the upper phase due to its lower density. Therefore, it can be used as a mobile phase in ascending mode, while changing to the heptane to limonene and keeping the same ratio, the opposite is true. Limonene is in the lower phase due to its higher density, making its use as a mobile phase in descending mode possible. At this composition, the low difference between the densities of the phases is also an advantage for CPC runs using limonene, which allows for the use of high rotational speed and reduced pressure drop compared to the classical systems. If besides the heptane limonene change, we change methanol to ethanol in the Arizona system, we can get a much more greener Arizona system, which further supports the previous observation that limonene can be used for the replacement of heptane. However, as in the Arizona system, ethyl acetate is also present. We must investigate its solubility in methanol and ethanol, where there is a significant difference in favor of ethanol. A major difference between the classical and this new green Arizona system is that limonene and ethanol are fully miscible, while in the case of heptane and methanol, they form a biophysics system at room temperature. Moreover, there is only a seemingly small difference between the polarity of methanol and ethanol, as we can see from the log P values in the table. However, this will mean that the distribution of ethanol will be much better in the between the upper and the lower phases in the green Arizona system than the distribution of methanol in the original one, resulting in more ethanol in the upper phases, raising the polarity of the system. We can say that for the same solutes, it will induce distribution, therefore separation differences between using the classical and the green Arizona system can occur. Another advantageous property of the green Arizona system is the low density differences of the phases for half of the green system compositions, which also will allow working with maximum centrifugal fields and therefore results in high stationary phase retention values, therefore better resolution. The substitution of chlorinated solvents is also a long desired goal in this chemical industry. The solvent selection guidelines mark almost all of them as hazardous or highly hazardous solvents. For example, the substitution of dichloromethane has not yet been fully implemented in organic synthesis or in chromatography. To replace dichloromethane, several investigations have been carried out. Besides dimethyl carbonate, methyl diacetyl ether, 2-methyl tetrahydrofuran, and cyclopentyl methyl ether was found to be a possible direct alternative. CPME has a low toxicity, high boiling point, and hydrophobicity, a low heat of vaporization, moderate density and viscosity, and a high surface tension, which makes it a possible and ideal choice for CPC separations as well. The CPME has also already been used successfully as a substitute for chloroform in high-performance liquid chromatography, and its application in CPC has also been reported as an alternative choice for methyl diacetyl ether, THF, and 2-methyl THF and dioxane. Also, if we are thinking about what can be in a green solvent system, water must come into mind as a first choice of green solvent because it is the safest and most environmentally friendly solvent, widely employed in various kinds of green separation processes. It is also the most polar solvent that we can choose. A special group of biphasic systems often used in liquid-liquid extractions is called aqueous two-phase systems, ATPSs, where the phase-forming components include two incompatible polymers, or a salt and a polymer dissolved in water, forming a biphasic system. This polymer is typically polyethylene glycol, which is used with phosphate buffers or sulfate buffers or dextrin or some kind of ionic liquid. One drawback of this system is that it is hard to recycle water after the separation. However, due to their high water content, these systems provide ideal environment for cells and organelles, therefore ideal for CPC separation of proteins, monoclonal antibodies, and virus-like particles. Within the confines of green chemistry, another group of compounds with suddenly growing interest is the group of so-called deep eutectic solvents, or when their components come from only natural sources, they are called natural deep eutectic solvents. These solvents can be characterized by a large depression of freezing point, which is usually higher than 150 Celsius degrees. The melting points of deep eutectic solvents are generally lower than 100 Celsius degrees. One of the most widely used components of this system is choline chloride, which meets the requirements of being cheap, biodegradable and non-toxic. And it is most widely used as the hydrogen bond acceptor in the system. And it can be paired with several possible hydrogen bond donors, such as urea, carboxylic acids, such as levulinic acid, different polyols, such as glycerol. As several poorly water-soluble drugs demonstrated the solvatation power of deep eutectic systems, they might act as a substitute for water and biphasic solvent systems used in CPC. It has been also shown that using the heptane, ethanol, and deep eutectic systems, using these biphases with similar density and density difference between the upper and lower phases can be created as compared to the classical HEMBAT systems. However, the viscosities of the phases will be significantly higher in this case, much more comparable to the ATPSs that I will just show you. As opposed to classical systems, we can also vary the different components of the deep eutectic systems to change the partition coefficients of the solutes, and we can adjust them this way to the preferred range of CPC without the change of the ratios of the components in the biphasic solvent system due to the different hydrogen bond donating and accepting affinity of the components of the DESs. Using this new type of biphasic systems, the successful separation of the hydrophobic model components, naringenin, retinol, beta-ionon, and alpha-tocopherol has been already successfully carried out using CPL. So we can conclude that these kind of systems can provide a promising alternative of water-free solvent systems with a polarity range facilitating mostly the separation of less polar and hydrophobic compounds. When we design a separation process and we design it to be sustainable and green, it is also desired to recycle most of the solvents whenever it is possible. To this way, several solutions have been developed, such as membrane filtration, ultrafiltration, dialysis and evaporation, and then density-based recirculation of the solvents. This density-based recirculation strategy has been developed by RotaChrom. In this automated system, the pure solvents are fed into the mixer settler unit, where the solvent system with the desired ratio of solvents is prepared. Then its composition is checked by simple density measurements. Then the prepared phases are stored in buffer tanks from where they are pumped into the CPC during the chromatographic run. After the separation is completed, the waste solvents and the solvents from the fractions are recirculated into the recycle skid and then back to the mixer sector. After the density-based readjustment of the solvent system, these recirculated solvents can be reused in the subsequent CPC cycles. Based on the purification project carried out at RotaChrom, almost 15 kilograms of crude steroid API could be purified with a really good average purity. And we needed 153 cycles for this. And in every cycle, 90% of the solvents could be recycled and reused. Every year, millions of tons of agricultural, industrial, and municipal waste is generated worldwide, and the waste treatment still poses a huge challenge for both the companies and the governments. Besides the conventional and physical chemical treatments, another alternative strategy to reach a zero waste future is the recycling, recovery, and then reuse of waste in the name of sustainable development. As a result of an environmental sound management of waste, the use of raw materials in manufacturing processes and the environmental impact of waste accumulation can be reused, and the valuable compounds can be recovered for the use in nutraceuticals, cosmetics, or pharmaceutics. A prime example of this is the recovery of hydroxytyrosol from table olive oil processing wastewater. This component is a phenolic compound in virgin olive oil, and it is a naturally derived antioxidative and remarkable free radical scavenging activity could be found, which can help in the prevention of reactive oxygen species mediated degenerative diseases. After the isolation of the phenolic compounds using non-ionic absorption resin, CPC could be successfully applied for the efficient and low-cost recovery of hydroxytyrosol. Another novel approach that can make CPC even greener is using the sugaring out strategy instead of salting out. This strategy might be used to fine-tune the solvent system in order to push the partition coefficients, the K values of the solutes, into the sweet spot of the CPC, which is the interval from approximately 0.5 to 2. In contrast to salting out, this sugaring out strategy uses simple sugars, which is more greener and environmentally friendly, and it does not produce some of the complications of salty corrosion. With the help of readily available sugars, we can quickly obtain a suitable solvent system with favorable K values. In the left graph, we can see the effect of increasing sucrose concentration on the K values of different solutes in a natural extract. With this approach, we can easily turn the not so favorable K values to ones that are in the sweet spot of the CPC separation. Also, in the top of the right corner, we can see that different kinds of sugars can affect the K values in a different way, which will give us even more opportunity and one more tool for the fine-tuning. By using a sugar gradient during the runs, the amount of the necessary sugars can also be decreased, and it has been found that the noise of the chromatograms also decreased in a classical HAMWAT system. So in general, we can say that sugaring out can be used as a strategy to provide a cheap and efficient method for optimizing the solvent system and simplifying the separation. So as a summary, we can see that we have several tools in our hands which can turn the application of CPC separation even more greener. We can select green solvent systems, or we can recycle the solvents after the separation runs. Also, the valorization of waste is one approach that must be considered, and we must look out for the novel approaches such as the sugaring out. Thank you for your kind attention. I am ready for your questions if there is any.
Thank you, Gergő. I hope that we managed to satisfy the information hunger, even those with a sweet tooth, with this sugaring out point in the last slide. We have a couple of questions. If you still have a question, there might be time to answer them. So there's a Q&A button. It should be on the bottom of your screen. So just press it and type away your question. So the first question I have here from one of the participants is just a general question. So if it's probably a non-chemist like myself, asking if they were to work with RotaChrom, are you going to be helping on arriving at a green solvent system, or how does one find a green solvent system for their specific separation?
Of course, if someone approaches us, we can do a solvent system screening, and if they ask for a green solvent system, then we can find for them the best applicant for that. So yes, it is possible to find a green solvent system. We can always take into consideration the needs of the customers.
Yeah. So what I would just add to this is that yes, Gergő, the research laboratory that Gergő works in, is specifically focused on feasibility studies and working on new methods. So they do come up with new methods and new solvent systems all the time. So yeah, just feel free to reach out to RotaChrom. The next question is about whether these techniques are applicable to other separation techniques such as HPLC. So I assume it's the question about the solvent systems, the alternative green solvents that you were presenting.
Yes, I think the answer to that question is yes, of course, because as I have shown you, the whole initiative was started by the pharmaceutical roundtable, and in the pharmaceutical industry, HPLC comes to mind as the first analytical technique. So yes, and it is a requirement there as well to become more greener and sustainable. So yes.
Thank you. The next question, thank you for typing that right now, actually, that I would like to ask is, first of all, thanks you for the nice talk, Gergő, and then inquires about how can we remove high boiling point solvents when contained in mobile phases?
Yes, this is a pretty tough question because in this case, evaporation can be energy consuming. So we must consider if it's worth it, or rather we can use another separation method for that, such as membrane filtration or ultrafiltration, or the ones I show you.
Okay, thank you for your answer. And for now, since we're a little short of time, okay, there are new questions coming in now. So let me just quickly read them. Okay, so the next question: what is the best green solvent system for cannabinoid separation and purification from hemp extract?
Well, if we say that there are several solvent systems for cannabinoid separation that use alkane, alcohol, water systems, as I have showed you, limonene can be used as a substitute for these alkanes. So I would think of a system using limonene, alcohol, and water.
Okay, thank you for that. We have a question about, I assume, these green solvents, if they work with SFCs as well. Sorry, no, it's I misread the question. So the question is, have you thought about working with SFCs as well?
Yeah, yes, we have thought about working, coupling CPC to SFCs, and others have also thought about that, and they tried that, but on the industrial scale, we have still work to do to make it possible. So it's quite hard from the engineering point of view, because we need to be able to contain those high pressures in the system, and that can be tough. So maybe in the future, I think it might be a real alternative or a real improvement of the systems. Yes.
Thank you very much. And the last question is just about a detail on slide 8. You mentioned that limonene was problematic. Is that from a regulatory perspective, or what perspective is it problematic from?
That is, yes, that was well, it was problematic from the point of view of the CAM 21 solvent selection guide. But as we can see, problematic is it's a much better category than the categories that the other solvents, hexane and heptane, were in, which were hazardous and highly hazardous. So I think that you can change to limonene instead of these hazardous solvents, and you get a still greener solution. And as we could see, these solvent selection guides were quite sometimes in contrast to each other. So I don't think that we can simply say that if one solvent selection guide says it's problematic, we shouldn't use that solvent.
Thank you very much. We will need to cut it after these two. We have two more questions. One that I will quickly take. A listener is asking if we have an application lab in the US. So our lab that Gergő is working in is in Europe, within the European Union. But we do have numerous partners in the United States that work with our equipment. Some are available for touring and watching how our equipment works and how does it look live. But we don't have ourselves application laboratory in the United States right now. And last question that we're going to answer today, because this is getting a little bit long now. Sorry, I just lost the question. One second. Is about the industrial. What is the maximum industrial scale possible? Is the question to Gergő.
Well, RotaChrom has the bigger machine. We call it ICPC, which have a 22 liters rotor, and in the case of that machine, we can purify kilograms of materials. So I think that is right now the maximum industrial scale for runs.
Thank you very much, Gergő. With that, we will close the Q&A section of today's webinar. And I would like to thank you for participating. A special thanks to Gergő for coming on and sharing all this information with us. If you would like to learn more, as I said in the beginning, please join us. I will be recording a podcast with Gergő, which should be available in the next week or so on your favorite podcast platform. We will continue to hold our webinars, but we will announce the next one very soon. So please, if you're not subscribed to our email list, go to the website rotachrom.com and subscribe to our email list or our newsletter, and you will get notification about our next webinar, which is going to be happening very soon. Thank you again once more for your attention and participation. If you have any further questions, you can always turn to us at sales@rotachrom.com. So shoot your questions there, and an application specialist will be in touch with you very soon. Thank you.